Hi guys, today we will be looking at a quick revision on carbohydrates. The three main groups of the carbohydrates that we're going to discuss are monosaccharides, which are monomers, disaccharides, which are made of two monomers, and polysaccharides, which are made of many monomers, joined together by the condensation reaction. So we will start with monosaccharides. As we know, mono stands for single, for one, so monosaccharides are monomers. So monomers are single units, and when monomers join together, they will join through the condensation reaction to form larger molecules such as disaccharides and polysaccharides. Two main groups, examples of the monosaccharides that we coming across in A levels are hexoses. Hexoses has 6 carbons. The examples of the hexoses are glucose. We've got two types of glucose, in fact, alpha glucose and beta glucose. They are isomers. So what does that mean? That means that they've got exactly the same chemical formula. So that stands for C6H12 and O6. However, the arrangements of the bonds, the arrangements of the atoms in the structure will be slightly different. So firstly, we will draw the alpha glucose. So, alpha glucose is a hexose, as we said, has 6 carbons. So let's label those carbons. This is carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, and carbon number 6. What makes alpha glucose differ from beta glucose? It's the fact that OH group, it's located at the bottom of carbon number 1. On the top, we can find hydrogen. In contrast, beta glucose at the position of carbon number 1 will have OH group on the top of it and hydrogen at the bottom. So those are main differences between alpha and beta glucose. It's really important you remember this because for the next groups of the when we look into polysaccharides, that will make a lot of difference in the structure of certain polysaccharides. Other examples of the hexoses that you should be aware of are, for instance, galactose. So we've got glucose and galactose, and we also have fructose. All three of those are examples of hexoses, hence the formula for all three of them stands as C6H12O6. Other group of the monosaccharides, now you will come across at A level, or you already have done some, are pentoses. Pentoses have 5 carbon molecules, and they are, for instance, found in the DNA and RNA molecule. We've got ribose, which you can find in the RNA nucleotide, and we've got deoxyribose, which then you can find in a DNA nucleotide. So briefly, those are examples of the monosaccharides, which obviously are monomers. They can join together through the condensation reaction to form larger molecules. If you are asked to provide the test that will allow you to recognize, to identify the monosaccharides, for instance glucose, in the food sample, we will use the test for reducing sugars. All monosaccharides are examples of the reducing sugars. So imagine here we've got your food sample with a glucose. To test for the presence of the reducing sugars, we will need to add the Benedicts. Benedicts, it's a blue reagent. And you will also need to heat the sample. So I put the red color underneath. So the simple method here for the reducing sugars is Benedicts and heat. Many students are forgetting about the idea of heating, so remember, to test for the presence of reducing sugars, you will add Benedicts and you will heat that solution. What will happen if the reducing sugars are present in a sample? You will observe the color change. So the color change, we'll go for the red one. That one suggests that there is a large amount of the reducing sugars. However, you could also get different color changes, which also gives you the positive results. We could get a green color. We could get yellow color. We could also get orange color. The way we've got different colors depends on the, of course, amount of the sugar present in your sample. But remember, the perfect model answer would be you would use the Benedict's test, and you will heat the solution, and the color will change from blue to red. Other group of the carbohydrates are disaccharides. So we're going to look into disaccharides now. Disaccharides, as the term stands for, di means two. So disaccharides are formed for the condensation reaction of two monosaccharides. There are three examples of the disaccharides that you need to be aware of. One of them is maltose. The other one that we've got is sucrose. And one more, lactose. We need to know what those disaccharides are formed from. So as we've said, they form from the condensation of two monosaccharides. Maltose, it's made of two alpha glucoses. Sucrose, it's formed by condensation reaction of glucose and fructose. And finally, lactose, it's made by condensation reaction of glucose and galactose. So what we need to obviously understand is the general formula of those disaccharides. So we did say that disaccharides are formed by the condensation reaction. So we're going to draw the condensation reaction for maltose using alpha glucose. So remember, we did talk about alpha glucose not long ago, and we did say that alpha glucose next to carbon number 1 has OH group on the bottom, hydrogen on the top. So I'm going to join those two together. Okay, so remember that was oxygen here, carbon number 1, 2, 3, 4, 5, and 6. Oxygen over there, carbon 1, 2, 3, 4, 5, and 6. Condensation reaction is the removal of water. So we want to remove water to form the bond. So water molecule will be removed from the OH groups. Following the condensation reaction, we are going to now achieve a disaccharide. So we got the oxygen that was left out here. This is our disaccharide. That's maltose. We also, don't forget, through the condensation reaction, remove the water molecule, hence the water is the product. And as the effect of the condensation reaction, we were able to join those two alpha glucoses together. They formed the bond now, and that bond, it's called glycosidic bond. Okay, so now we wonder what is actually the formula of the disaccharides. So let's remind ourselves what have we actually done here. So we carry out the condensation reaction of two monosaccharides, of two glucoses. The formula of the glucose is C6H12O6. So we've joined two of those. However, we removed water as the effect of condensation reaction. So if we do maths here, we come up to the conclusion that the general formula for disaccharides stands as C12H22O11 if we join two hexoses. Disaccharides, except for the maltose, are examples of the non-reducing sugars. Hence, the testing for the presence of them in the food sample will be slightly different. So, the method used for the non-reducing sugars, it's called is slightly different than the method for reducing sugars. So we will start with exactly the same approach as we've done it for the reducing sugars. So we will use Benedict's and we will heat that solution as well. However, by doing so remember, this was our sample, Benedict's reagent, it's blue. We heat the sample. However, we're not achieving positive results yet. So in here, our sample will still remain blue. There is no color change. So what we need to do next then, we need to again heat our sample, but now with the hydrochloric acid. So to our sample, now we are going to add hydrochloric acid. So we got the Benedict's and we're adding hydrochloric acid. The reason why we're doing this is to break the glycosidic bond. So remember, that was the bond located between carbon number 1 of the first glucose and carbon number 4 of the second glucose. Okay, so carbon 1, carbon 4. So obviously by adding hydrofluoric acid, we have changed the pH now of our solution, becomes acid. So now we want to neutralize our solution. And to do so, we're going to use sodium hydrogen carbonate. Once we follow all those steps, we are going to now come back to our first step of the methods. So we're going to now try and use Benedict's and of course heat our solution. Now we are hoping, well, we should predict and be confident that this is going to give us positive results because we broke the glycosidic bond and we've got monosaccharides, and we know that monosaccharides are reducing sugars. So following this method, we are again going to add Benedict's and heat our solution. By doing so, we will now achieve positive results. So the red brick color, so the solution will change from blue to red. So that is a method for non-reducing sugar, and the most common mistake students are making is forgetting about the idea of heat. So don't make those mistakes. So that's disaccharides and the methods of identifying the presence of the non-reducing sugars in the food sample. The last group of the carbohydrates that we need to be aware of are polysaccharides. Poly, as the word stands for, means many. So there will be many monosaccharides joined by the condensation reaction, which is removal of water that allows the formation of the glycosidic bonds. So in terms of our specification, we need to be able to talk about three polysaccharides. Those polysaccharides are starch. Starch, of course, it's found in plants. It's a storage material. And starch is made of the repeated monomers of alpha glucose. So as we remember, alpha glucose joined by the condensation reaction, forming one 4-glycosidic bond. Starch molecule is not as branched, hence we will have mainly 1,4-glycosidic bonds. Obviously, it's a long polysaccharide, hence allowed a lot of information to be stored. Another molecule, polysaccharide, that we need to be able to recognize is glycogen. Glycogen is found, of course, in our cells, and it's a storage material. Similarly as starch, glycogen is found of the alpha glucose. However, glycogen is more branched, means it gives more spaces for the enzymes to work on, and so in glycogen we will have 1,4 but also 1,6 glycosidic bonds. So 1,4-glycosidic bond obviously was formed between carbon number 1 and carbon number 4. However, 1,6-glycosidic bond will be formed between carbon number 1 and carbon number 6. So if I will draw here another glucose, Okay, this is carbon number 1. I want to now make a glycosidic bond between 1 and 6. Remember, 6 was the one over there. That's then the location of the glycosidic bond between carbon number 1 and 6 over there, which makes it more branched, so more storage material, so more ends for the enzymes to work to hydrolyze glycogen into glucose, which then can be used for respiration. And this is 1,4-glycosidic bond, which obviously makes the structure less branched, and that's starch. And the final polysaccharide that you need to be able to talk about is cellulose. Cellulose again found in plants. So cellulose makes the cell wall. This is the polysaccharide that it's made of beta glucose. It will have 1,4-glycosidic bonds. However, in contrast to starch and glycogen, cellulose is a straight molecule. So, how is it possible to maintain the straight molecule having 1,4-glycosidic bonds? Remember, beta-glucose differs from alpha-glucose. Beta-glucose has the OH group on the top of carbon number 1. So the position, obviously, of the glycosidic bond will slightly differ. So let's try and draw it out. So we've got here beta-glucose forming the glycosidic bond. In that way. So we'll have a look at this in a second. Why the oxygens are in different positions. The reason why this glycosidic bond sometimes is on the top, sometimes is on the bottom, it's the fact that the beta glucose rotates by 180 degrees. Hence, when you look at the past paper questions, you can really often see the chains for starch, glycogen and cellulose, and you probably know only once for why the glycosidic bond, sometimes it's formed on the top, sometimes on the bottom. So remember that glucose, beta glucose, flips by 180 degrees, allowing such a certain assortment of the bonds and position of the glucoses in the chain. The reason behind that is because we've said that cellulose makes the plant's cell walls. So it has to be a straight and strong molecule. So imagine now here we've got many chains of beta glucose. So the single lines are the chains of beta glucoses, so the cellulose. Beta glucoses then arrange themselves into those chains, forming microfibrils. Those microfibrils are then supported together by the presence of the hydrogen bonds. So that structure provides then the strength and support and allows the chain, obviously here what we've discussed previously, to be straight, allowing formation of the plant cell wall. In terms of the tests that we need to be able to recall for the presence of the polysaccharides in the food samples, it's only the one that we need to really know here. It's the test for the presence of starch. The test that we will use here is the iodine. And the positive results, change of color for the iodine test for starch, it will be dark blue, black. So if you've done the practical at school, you will see that your sample, food sample that contains here starch, after you've added iodine, is going to turn really dark. So dark blue, black color. So that's everything in terms of the structure and the function of the carbohydrates. So we discussed monosaccharides, disaccharides, and polysaccharides. Now, plenty model answers out there to link the structure to the function. Make sure you will do now many past paper questions to be able to apply the AO1 knowledge.